welcome to this video lecture. Now let us begin this video lecture with a brief definition of free radicals. Now there are two characteristics that define what free radicals are. Okay? The first is they have to be atoms that have unpaired electrons in their outermost orbital. And second, they're capable of independent existence, hence the term free, or free radicals for that matter. Now, as you will learn in your higher years, you will see that free radicals are largely involved in the pathogenesis of a variety of conditions, okay? The partial list of which is shown here. Now, radicals, whereof free radicals are a subset, are highly reactive species on account of that unpaired valence electron, okay? Which invariably initiates chain reactions by extracting an electron from a neighboring healthy molecule in order to complete their own orbitals, right? Now, that healthy molecule, in return, now has an unpaired electron and becomes itself a radical, which is capable of propagating this chain reaction of electron extraction, ultimately leading to cellular and DNA damage. Now, conventionally, antioxidants provide these radicals with electrons to terminate this chain reaction. Technically, transition metals such as iron, copper, and molybdenum, which act as inorganic prosthetic groups in many proteins, also have unpaired electrons in their outer shell. But relative to radicals, however, they are more stable on account of their complexing with proteins, and they do not initiate chain reactions. Okay? They can, however, be targets of free radicals, causing them to change from a higher to a lower oxidation state, and this can frequently affect the protein's function. Now, believe it or not, the most stable form of molecular oxygen, or O2, actually has two unpaired electrons occupying two molecular orbitals, as you can see on the left of the image. Now, these electrons have the same spin and are unable to form a bond, thus making oxygen what's called a biradical, right? Now, this biradical form of oxygen is referred to as a triplet oxygen. Now, singlet oxygen, on the other hand, which is on the image here on the right, has higher energy and is much more reactive than triplet oxygen. The air we breathe basically contains triplet oxygen, and this biradical is what drives the biochemical processes of aerobic systems. Now, the biradical nature of oxygen allows it to drive many oxidation reactions ranging from, say, the spoilage of food to oxidative damage in cells. Now, for that matter, free radicals are mostly products of oxygen metabolism, wherein the biradical oxygen accepts single electrons from molecules and effectively gets converted into reactive oxygen species, the most commonly described being superoxide, hydrogen peroxide, and hydroxyl radical. Now, metal-containing enzymes are typically the ones that act as catalysts in ROS formation. Now, basically, ROS generation is a natural part of everyday life. They are but accidental products of both non-enzymatic and enzymatic reactions. Okay? The electrons that get added on to the biradical oxygen are generally derived from reduced electron carriers of the electron transport chain, mainly your NADH. Okay? Now, 3 to 5% of the molecular oxygen we consume gets converted to free radicals. Now, here's the reaction involving formation of superoxide, hydrogen peroxide, and hydroxyl radical. Now, of note, ultraviolet radiation and pollutants can increase ROS formation. Now, besides normal aerobic metabolism, ultraviolet light, and pollutants, ionizing radiation as well as systemic inflammation that occurs during infection also contribute to free radical formation. Now, as was mentioned earlier, this ultimately leads to DNA damage when free radical chain reactions are allowed to propagate. Okay? Hence, their involvement in the pathogenesis of a variety of non-communicable diseases, which I've shown a few slides ago. Now, you've previously learned under bioenergetics about semiquinone formation during reoxidation of the prosthetic groups of flavin-containing enzymes, right? Now, semiquinone is basically a free radical that results from the removal of one hydrogen atom with its electron. Now, free radical intermediates, which are formed during normal metabolism, can themselves get accidentally released from enzyme active sites before their reduction is completed, right? As mentioned earlier, metal-containing enzymes are typically the ones that act as catalysts in ROS formation. Now, you can also generate free radicals via hydroxylation reactions that occur in cytochrome P450 during the detoxification of xenobiotics, which will be our next video lecture. Now, similarly, P450 enzymes can be induced as well by substances such as alcohol, certain drugs or medications, and certain chemical toxicants such as carbon tetrachloride or tetrachloromethane. Okay. Now, the complex biochemical processes that are involved in steroid hormone synthesis can also be sources of free radicals, since these processes involve cytochrome P450 enzymes. Now, the synthesis of eicosanoids, which was discussed previously under lipid metabolism, involve metabolic pathways that enzymatically form lipid peroxidases as intermediates. Now, recall that lipid peroxidases are intermediates that are themselves radicals, right? Now, xanthine oxidase, which is an enzyme that catalyzes the oxidation of hypoxanthine to xanthine, as well as its further oxidation to uric acid, can likewise generate ROS. Now, the reaction catalyzed by this enzyme inadvertently reduces molecular oxygen to superoxide or hydrogen peroxide as well. And this typically happens during what's called ischemia reperfusion or reoxygenation injury, which is defined as the damage tissues sustain when blood supply returns after a period of ischemia or lack of oxygen. Now, ROS formation is believed to drive this damage. Now, this injury is associated with serious clinical manifestations such as acute heart failure, cardiac arrhythmias, cerebral dysfunction, kidney failure, gastrointestinal dysfunction, systemic inflammatory response syndrome or SIRS, and multi-organ dysfunction. Now, molecular oxygen carried by RBCs or erythrocytes can also accept an electron during the conversion of hemoglobin to methemoglobin, as depicted here in this reaction. This then generates superoxide free radicals in the process. Reactive oxygen species can likewise be produced by phagocytic cells, right? And this involves the formation of hypochlorous acid. Now, ROS formation, on the other hand, becomes deliberate during inflammatory responses, right? Now, when stimulated by bacteria, neutrophils exhibit what's called a respiratory burst, and they produce superoxide in a reaction catalyzed by NADPH oxidase. So here's the reaction. Basically, it is this production or generation of superoxide that is one of the several ways in which neutrophils are able to kill bacteria. Now, as mentioned earlier, reactive oxygen species or ROS formation requires the help of a catalyst, typically an enzyme with a metal ion prosthetic group. Okay? Now, basically, their formation is not a thermodynamically favorable reaction, requiring a moderately strong reducing agent that can donate single electrons. With that agent, typically a partially reoxidized coenzyme Q, or any semiquinone for that matter, right? Now, ROS, in turn, extract electrons, usually as hydrogen atoms, from other compounds to complete their own orbitals. Now, the reactivity and destructiveness of individual reactive oxygen species varies, with hydroxyl radicals being the most destructive and hydrogen peroxide being the least destructive. Now, strictly speaking, hydrogen peroxide is not a radical, but rather a weak oxidizing agent, but is still considered a reactive oxygen species because of this reason. Okay? Now, there are specific pathways by which the less destructive ROS can transform to more highly reactive ROS in the presence of iron ions. So these are the Fenton and Haber-Weiss reactions. Okay? The former is actually a non-enzymatic reaction wherein the ferrous ion is able to transform hydrogen peroxide to hydroxyl radical. The latter reaction, on the other hand, involves the reaction of superoxide and hydrogen peroxide to generate hydroxyl radicals. Now, this reaction makes use of iron as catalyst. So we can actually summarize this entire process into four chemical reaction equations. The first one here is the Fenton reaction, where the ferrous ion transforms hydrogen peroxide to hydroxyl radical. Now, the hydroxyl ion generated can react with another hydrogen peroxide molecule to produce superoxide. And that superoxide, in turn, can reduce the ferric ion from earlier back to the ferrous state, right? Now, if we summarize these first three reactions, we get what is called the Haber-Weiss reaction, which, as explained earlier, involves the reaction of superoxide and hydrogen peroxide to generate hydroxyl radicals. Now, even though iron is cancelled out in this equation, as you can see, it doesn't mean it's not there, right? As mentioned earlier, iron in this case acts as a catalyst. Now, as will be explained in the next several slides, we will learn that reactive oxygen species affect and potentially damage a number of cellular components, particularly membrane lipids, cellular proteins, and DNA. Now, free radicals can react with the polyunsaturated fatty acids found mainly in membranes via a process called lipid peroxidation, which is really a chain reaction that produces a continuous supply of more free radicals. Now, this chain reaction can lead to loss of membrane integrity, affecting not only the plasma membrane but also the mitochondrial membrane. Now, with destruction of the mitochondria, obviously, the function of the respiratory chain and the production of ATP is severely undermined. Now, as what I just mentioned just now, lipid peroxidation is a chain reaction involving polyunsaturated fatty acids. Now, in this free radical generating process, malondialdehyde is generated, right? Now, this compound in itself can also covalently react with DNA, proteins, and lipids, forming what are called adducts that can produce more cellular damage. Now, by the way, in biochemistry, an adduct refers to the product of an addition reaction between two compounds. Now, malondialdehyde appears in blood and urine, something that some specialized centers use as an indicator of free radical damage. Now, this figure here summarizes the process of lipid peroxidation. Initiation commences when a hydroxyl radical reacts with an unsaturated fatty acid, forming a lipid radical, which is designated here as L dot. Now, with propagation, the process of lipid peroxidation is continued in the presence of molecular oxygen to form a lipid peroxyl radical, designated here as LOO dot. Now, recall from earlier that molecular oxygen is a biradical, right? Now, the action of a lipid peroxyl radical with another, or rather with other unsaturated fatty acids, as what could happen in cell membranes, lead to the formation of lipid peroxide, which is designated here as LOOH, or more lipid radicals for that matter, thereby propagating the process of lipid peroxidation, right? Now, the process is terminated when lipid radicals and lipid peroxyl radicals react to form lipid peroxides and fatty acids. Now, lipid radicals and lipid peroxyl radicals can also react with antioxidant vitamins such as vitamin E, and the propagation process is terminated. Now, of note, lipid peroxides are further degraded into malondialdehyde and other lipid peroxide degradation products. So, I already talked about malondialdehyde earlier. Now, normal low-density lipoprotein or LDL metabolism can likewise be altered by reactive oxygen species. Okay? Now, lipid peroxidation causes LDL to undergo chemical modification, particularly oxidation, leading to conformationally abnormal oxidized LDL. Okay? Now, this oxidized LDL are no longer recognized by your LDL receptors in the liver. So, as a consequence, they are not cleared from the bloodstream. Okay? Now, these oxidized LDL are instead taken up by macrophage scavenger receptors via the so-called scavenger pathway as they freely enter the vessel wall and accumulate underneath the vessel endothelium, right? So take note that LDL is not normally taken up by the vessel wall, right? Now, macrophages in the vicinity then internalize the oxidized LDL, eventually becoming lipid engorged with high content of unesterified cholesterol. Now, these macrophages effectively turn into what we call foam cells, which then trigger the release of cytokines and growth factors that create a milieu of microinflammation in the endothelium. Now, this makes it conducive for atherosclerosis to form in the endothelium, right? Now, the lipid-engorged macrophages that infiltrate the blood vessel eventually die, contributing to the atherosclerotic process. Now, for those of you who find it easier to use concept maps, here's a flowchart to which Dr. RVSN Sarma is credited. And this summarizes the discussion from the previous slide. Now, how do reactive oxygen species affect cellular proteins? So, ROS reactions with proteins may cause the latter to fragment. Now, similarly, amino acid R groups may likewise cross-link, thereby preventing physiologic degradation of involved proteins. Now, the amino acids proline, histidine, arginine, cysteine, and methionine are particularly susceptible. Now, one very important instance of the harmful effects of cross-linkage would be with the cysteine-containing proteins, a popular example of which is glutathione. The thiol group of the cysteine residue in reduced glutathione is able to provide electrons or reducing equivalents to complete the incomplete orbitals of radicals, right? Now, this property of glutathione affords it its antioxidant properties. Now, in the healthy state, more than 90% of the total glutathione pool exists in the reduced form, with the remainder in the oxidized form, if you could still remember from previous lectures. Now, the reduced to oxidized glutathione ratio within cells is an important measure of cellular oxidative stress. If there's more of the oxidized form relative to the reduced form, this is indicative of greater oxidative stress, right? Moreover, the thiol groups of oxidized glutathione are engaged in cross-linkage, right? And therefore do not have any antioxidative properties. Additionally, ROS reactions with proteins can increase their susceptibility to proteolytic digestion. And reactive oxygen species can also inflict damage on DNA and RNA. Now, base alterations and strand breaks can result. Now, such forms of damage to genetic material have been implicated in the development of cancer, right? Now, here's the stick representation of the oxidized form of glutathione. Note that the thiol groups are engaged in cross-linkage and therefore do not have any antioxidative properties that reduced glutathione has. Now, hopefully you can still recall this from your lecture on the pentose phosphate pathway. Now, reduced glutathione is restored from oxidized glutathione via the enzyme glutathione reductase in the presence of NADPH. Now, the oxidized form of NADPH, which is NADP+, is in turn restored back to its reduced form by glucose 6-phosphate dehydrogenase or G6PD, the first step in the oxidative phase of the pentose phosphate pathway. Now, like ROS, nitric oxide or NO is also produced naturally by our bodies. It is also an oxygen-containing free radical, that is, it contains a single unpaired electron. Okay? Now, nitric oxide is interesting in that it can act as a double-edged sword. This means that at low concentrations, it functions in neurotransmission and signal transduction. At high concentrations, however, it can combine with the molecular oxygen or superoxide to form what we call reactive nitrogen-oxygen species or RNOS, which have been implicated in a number of neurodegenerative diseases such as Parkinson's disease and in chronic inflammatory diseases such as your rheumatoid arthritis. Now, nitric oxide is produced by a group of enzymes called nitric oxide synthase or NOS, which have three isoforms, namely your neuronal or NNOS, endothelial or ENOS, and inducible or INOS. Okay? Now, the neuronal and endothelial isoforms are constitutively expressed in mammalian cells, particularly in the central and peripheral nervous system and the vasculature respectively. Now, in the case of neuronal NOS, interaction of certain neurotransmitters or extracellular signals such as acetylcholine, serotonin, thrombin, and bradykinin to their cellular receptors affect influx of calcium ions intracellularly. Now, the calcium ions bind to a multifunctional calcium binding protein, which we call calmodulin or CAM, which in turn activates neuronal NOS in the nervous system and endothelial NOS in the vasculature. Okay? Now, unlike neuronal NOS, endothelial NOS, however, when it's activated, also can be independent of calcium ions, meaning endothelial NOS can also get activated in response to vascular shear stress, which is the frictional force generated by blood flow in the endothelium. Okay? So basically, endothelial NOS activation can also be independent of calcium ions, unlike your neuronal NOS. Okay? Now, NOS makes use of L-arginine as substrate. Now, in the presence of NADPH and tetrahydrobiopterin or BH4, it gets converted to citrulline, releasing nitric oxide as a byproduct. Now, nitric oxide then binds to endothelial guanylyl cyclase. Now, hopefully you can still remember this from your signal transduction lectures. Now, this enzyme, guanylyl cyclase, as you can recall, converts GTP to cyclic GMP or cGMP, which induces smooth muscle relaxation, leading to vasodilation. Now, as mentioned earlier, nitric oxide can react with molecular oxygen or superoxide to form your RNOS. Okay? Now, when nitric oxide reacts with molecular oxygen and other nitric oxide molecules, nitrogen trioxide forms. Now, nitrogen trioxide is what's called a nitrosating agent, like nitrates that are used as food preservatives. Now, nitrosation is the process of converting amines, which are contained in organic materials such as proteins in foods, into nitrosamines, right? Now, nitrosamines have been implicated in a variety of health conditions, the most described of which is cancer, right? Now, such compounds were found to experimentally induce cancers in laboratory animal models and may increase the risk of certain types of cancers in humans, particularly gastric cancer, right? Now, nitrogen trioxide can also nitrosylate sulfhydryl and similarly reactive groups in cell components, leading to interference in proper functioning of the affected proteins or lipids. Okay? Now, reaction of nitric oxide with superoxide, on the other hand, leads to the formation of peroxynitrite, which is a strong oxidizing agent. Okay? Now, although peroxynitrite is not technically a free radical, okay, its strong oxidizing properties allow it to potentially damage a wide range of biological molecules, and that includes your DNA and proteins. Okay? Now, at physiologic pH, peroxynitrite exists in its conjugate acid form, which we call peroxynitrous acid. Okay? And this is a strong 2-electron oxidant and has a longer half-life than your superoxide. Now, peroxynitrous acid is an unstable species which can permeate across hydrophobic biocompartments, and by that I'm referring to your cell membranes, where it decays into nitrogen dioxide and hydroxyl radical, both of which initiate lipid peroxidation that I discussed earlier. Okay? Of note, nitrogen dioxide is a toxic agent that you find in smog, cigarette smoke, organic smoke, and by that I refer to smoke coming from forest fires, as well as automobile exhausts. Okay? Now, peroxynitrous acid can also decay into hydroxyl radical and nitronium ion, which is a nitrating agent. Okay? Now, lastly, peroxynitrous acid can decompose to nitrate ion, the only nitrogen-oxygen containing compound that is relatively non-toxic. Now, nitric oxide can also exert their toxic effects through combination with iron-containing compounds that also have single electrons, namely, number 1, your sulfur or iron-sulfur centers rather, which are found in complex 1 and complex 3 of the electron transport chain, as well as in the Krebs cycle enzyme aconitase. And number 2, your iron heme proteins, such as your hemoglobin in blood, as well as the cytochromes in the electron transport chain. Now, as can be seen in these examples, nitric oxide can cause direct inhibition of cellular respiration when in sufficient amounts, right? Now, nitric oxide, when present in low concentrations, usually cause little to negligible damage because iron heme compounds are present in excess capacity. Okay? Now, small amounts of nitric oxide become toxic, however, during ischemia or inflammation, or in the presence of hereditary diseases of oxidative phosphorylation. A good example of which would be your MELAS syndrome. Okay? Let's move on now to the topic of respiratory burst. Now, respiratory burst, which is sometimes called oxidative burst, is the rapid increase in production of and release of your ROS and RNOS, okay, which typically happens during phagocytosis of microbes. Now, it is an essential part of one's innate immunity, okay, enabling phagocytic cells such as neutrophils, eosinophils, monocytes, and macrophages to ingest and eliminate invading pathogens, okay. Now, as the name entails, or as the name implies, rather, respiratory burst involves rapid consumption of molecular oxygen and lasts only 30 to 60 minutes, supposedly. Okay? Now, two enzymes are involved in the respiratory burst. The first is your NADPH oxidase, which catalyzes the transfer of an electron from NADPH to molecular oxygen to form your superoxide. Now, this reaction occurs in the outer side of the plasma membrane and basically initiates the respiratory burst process with the production of your superoxide. Okay? Now, hydrogen peroxide is also produced from superoxide either spontaneously or via the enzyme called superoxide dismutase, which I'll be discussing later on. Now, through the Fenton reaction, which I've already discussed earlier, hydrogen peroxide can be converted to hydroxyl radicals. Okay? Now, with invagination of the cell membrane during phagocytosis, superoxide is released into the vacuole space containing the ingested microbe. Now, superoxide can also react with nitric oxide to form your RNOS, which eventually attacks the membranes of the phagocytosed microbes, leading to their destruction. Okay? Now, the other enzyme involved in the respiratory burst is myeloperoxidase. Now, this enzyme, predominantly in neutrophils and monocytes, catalyzes the formation of your hypochlorous acid, okay, which is a powerful agent that destroys bacteria via halogenation and oxidation. Well, since chloride is a halogen, okay. Now, hypochlorous acid also oxidizes a variety of iron and sulfur-containing groups, such as your iron-sulfur centers, your iron-heme centers, your ferredoxin and methionine. And this gives off a greenish color, which is often observed in phlegm or respiratory secretions of patients combating or fighting bacterial respiratory infections. Okay? Now, here's the graphic representation of my discussion from the previous slide, okay, which summarizes the actions of the two key enzymes NADPH oxidase and myeloperoxidase. Now, take note of how the ROS and RNOS are produced here. Now, on the most part, respiratory burst spontaneously subsides following successful containment of the infection, leading to complete recovery of the host. Okay? However, it can act as a double-edged sword as well. Now, sometimes the inflammatory response, particularly during sepsis or sepsis-like syndromes, can become overwhelming that it leads to systemic vasodilation and capillary leakage, both of which lower effective intravascular volume and maldistribution of blood flow, eventually leading to hypoperfusion of peripheral tissues and organs. Okay? Now, due to hypoperfusion coupled with uncontained infection, the inflammatory response becomes progressive instead of resolving, leading to a systemic inflammatory response syndrome or SIRS, where there is an excess of ROS and inflammatory mediators and cytokines, such as your interleukins and tumor necrosis factor. Okay? If not addressed in time with the proper antibiotic treatment, the systemic inflammatory response syndrome or SIRS can lead to multi-organ dysfunction and failure, and ultimately the death of the patient. Alright, now this diagram here is rather simplistic, but in normal situations, pro-oxidant molecules are generally countered by the effect of antioxidant molecules so as to mitigate any excessive oxidative stress to the cells and tissues to avoid their damage. Okay? Now, in the following slides, I'll be discussing our cellular defenses against oxygen toxicity or free radical toxicity. So the first group of cellular defenses include antioxidant scavenging enzymes. Now, this group includes the enzymes superoxide dismutase, catalase, glutathione peroxidase, and glutathione reductase. Now, as the name of this group implies, they are enzymes that manifest their antioxidant properties by scavenging free radicals, right? Of note, most enzymes that produce and degrade free radicals are confined in your peroxisomes, okay? Now, the second group involves non-enzymatic antioxidants, also referred as non-enzymatic free radical scavengers. Now, this group includes your polyphenols and antioxidant vitamins, namely your vitamin C, vitamin E, and your beta-carotene. Now, as the name of this group implies, the compounds included in this group of antioxidant cellular defenses, quote-unquote, trap and stabilize free radicals via the extended conjugated double bond system of their molecular structure. Okay, we will be talking about that later. Now, recall what I said earlier that your ROS formation is not a thermodynamically favorable reaction. Okay? Now, this process requires the help of a catalyst, which is typically an enzyme with a metal ion prosthetic group. Alternatively, the presence of a moderately strong reducing agent that can donate single electrons from molecular oxygen can also favor the formation of your ROS, which I already discussed earlier. Remember that I mentioned partially reoxidized coenzyme Q and the semiquinone molecule as typical examples of such strong reducing agents. Now, the transfer of a single electron that is involved in this process generates a potentially damaging superoxide anion free radical, okay, which can give rise to free radical chain reactions, thereby amplifying its destructive effects. Okay? Now, additionally, superoxide can reduce oxidized cytochrome C in the electron transport chain. This basically leads to the disruption of the function of cytochrome C as an electron carrier in the ETC. Now, fortunately for us, superoxide is removed by this enzyme called superoxide dismutase, which catalyzes the conversion of superoxide to molecular oxygen and hydrogen peroxide. Now, this conversion by superoxide dismutase is what is called dismutation. So, superoxide dismutase, which occurs in 3 isoenzyme forms, as you can see here, primarily protects aerobic organisms against the deleterious effects of superoxide. Now, if you go back to the figure in the previous slide, note that the hydrogen peroxide that is formed from superoxide can still potentially oxidize and reduce iron in cytochromes, which makes this compound still toxic technically. Okay? Thus, hydrogen peroxide has to be converted into another non-toxic compound, and this would involve your catalases. Okay? So these enzymes reduce hydrogen peroxide to water, thereby preventing the formation of hydroxyl radicals by way of the Fenton or Haber-Weiss reactions that I've already discussed earlier on. Now, another antioxidant scavenging enzyme is glutathione peroxidase. Now, this is a selenium-containing enzyme and is a major means for removing hydrogen peroxides as well as lipid peroxides produced outside of the peroxisome, thereby protecting the organism from oxidative damage. Okay? Now, this enzyme is likewise capable of reducing peroxide radicals to alcohol and oxygen, as you can see in the left part of the illustration. Okay? Now, in order to detoxify peroxide and peroxide radicals, so this enzyme obtains its electrons or reducing equivalents from reduced glutathione, which gets converted to oxidized glutathione in the process. Now, glutathione reductase, which is an FAD-containing enzyme, is the one responsible for restoring oxidized glutathione back to the reduced glutathione form by catalyzing the transfer of electrons from NADPH to the disulfide bond of oxidized glutathione. Now, the oxidized form of NADPH, which is your NADP+, is in turn restored back to its reduced form by glucose 6-phosphate dehydrogenase or G6PD, which I discussed earlier. So as you can recall, the enzyme catalyzed by, the step catalyzed by the enzyme G6PD is the first step in the oxidative phase of the pentose phosphate pathway. Okay. So we leave now the topic of antioxidant scavenging enzymes and move on to free radical scavengers. Now recall from earlier that I mentioned that these compounds quote unquote trap and stabilize free radicals via the extended conjugated double bond system of their molecular structure. Now this figure here shows the general structure of the free radical scavengers, which contain that so-called extended conjugated double bond system, which is in green here in this figure. Okay. Now let's start off with polyphenols. So polyphenols are compounds that contain several hydroxyl groups in two spatially separate aromatic ring systems. So I'll show you a figure of that later. Now they occur naturally in fruits, vegetables, tea, coffee, and certain beers and wine. Now, polyphenols are generally present in plants to protect them against ultraviolet radiation or aggression by plant pathogens. Now, they generally function by inhibiting enzymes responsible for superoxide production, such as your xanthine oxidase, which was discussed earlier. Now, they can also effectively chelate iron and copper ions so that they cannot participate in the radical generating Fenton reaction. They likewise have the ability to stabilize radicals by either donating electrons to superoxide or lipid peroxy radicals or directly complexing with free radicals. Okay, so once they do that, then they terminate the chain reaction. Okay. Now, I mentioned earlier about polyphenols having several hydroxyl groups in two spatially separate aromatic ring systems. Okay? Now, I'd like you to focus your attention on the uppermost panel of this figure. So, the areas circled in pink show features that are critical for efficient antioxidant function, right? Now, notice well how these hydroxyl groups are found in separate aromatic ring systems. Now, you can pause this video so that you can read the rest of the description here. Now, here are some examples of polyphenol compounds. They include curcumin, found in turmeric, ferulic acid, found in rice, wheat, barley, oats, coffee, tomatoes, and citrus fruit, epigallocatechin gallate, or EGCG, found in green tea, and resveratrol, or resveratrol for that matter, found in grapes and wine. Okay? Next, we have vitamins E and C. Now, vitamin E, the most potent of which is your alpha tocopherol, functions to protect against lipid peroxidation, which frequently occurs in cell membranes, as was discussed earlier. Okay? Now, from a previous figure earlier on in this video lecture, you can see that vitamin E reduces lipid peroxides to fatty acids, converting vitamin E to a tocopheroxyl radical in the process. Now, this tocopheroxyl radical is eventually converted back to the reduced form of vitamin E via vitamin C by donating electrons in the redox cycle. Now, this is generally observed on cell surfaces as well as lipoproteins. Now, this is just to show you what I was talking about regarding vitamin E and lipid peroxides. Now let's go into a little more detail with regards to vitamins E and C. Now, as was discussed previously, vitamin E reduces lipid peroxides to fatty acids. Now, this causes vitamin E to, in return, get converted to a tocopheroxyl radical, which is thankfully stable, meaning it does not easily partake in the propagation of radical formation. Now, this tocopheroxyl radical is eventually converted back to the reduced form of vitamin E by vitamin C. Now, it does this by donation of electrons in the redox cycle, thereby causing vitamin C to get converted in the process to what is called a monodihydroascorbate, which is the radical form of vitamin C. Now, this monodihydroascorbate eventually undergoes either an enzymatic reaction, or rather enzymatic reduction back to ascorbate, or a non-enzymatic reaction with another monodihydroascorbate molecule to form one ascorbate or dihydroascorbate molecule. Okay? Now, let's tie all the concepts up together here in this figure. So we have here vitamin E converting lipid peroxyl radicals that form in the cell membrane to lipid peroxides. The tocopheroxyl radical, which is basically your oxidized vitamin E, which forms in the process, is eventually converted back to its reduced form by vitamin C. And vitamin C then gets oxidized in the process. Okay? Now take note as well that reduced glutathione can also convert tocopheroxyl radicals back to reduced vitamin E, with it getting converted to oxidized glutathione in the process. Okay? Now oxidized glutathione then has to be converted back to its reduced form via the action of glutathione reductase, which I already discussed earlier on. Now, in the cytosol, the peroxides that accumulate are acted upon by catalase to convert them to non-toxic compounds. Now, from earlier discussion as well, superoxide dismutase converts the highly toxic superoxide radical to hydrogen peroxide, thereby adding to the peroxide pool, which eventually catalase acts on to convert into non-toxic compounds. Let's move on now to carotenoids. So carotenoids are yellow, orange, and red pigments produced by certain plants, algae, and fungi, as well as several bacteria. They likewise give the characteristic color to carrots, pumpkins, corn, and tomatoes. Now, beta-carotene is the antioxidant carotenoid precursor to vitamin A or retinol, and is a strongly red-orange colored pigment that is abundant in carrots and certain fruits and vegetables. Now, other members of this group include lutein and zeaxanthin, both of which are predominant carotenoids that accumulate in the retina of the eye. And they have been found in a 2017 meta-analysis to delay the progression of age-related macular degeneration. Now, here are the forest plots in that meta-analysis in case you're interested. Okay? Now, prior studies have observed correlations between health benefits and diets high in fruits and vegetables containing carotenoids. And because of that, several researchers have hypothesized that perhaps such compounds may slow progression of cancer, atherosclerosis, and degenerative diseases. Okay? Now, in a 2008 meta-analysis performed by Tanvetyanan and colleagues, however, it was found that among current smokers, beta-carotene appeared to increase rather than decrease their odds for getting lung cancer, and this is by a factor of 1.24. Now, these results seem rather surprising at first glance because since beta-carotene is considered to be an antioxidant, you're expecting it to be protective rather than harmful. Now, a possible explanation for these findings can be traced to laboratory studies showing beta-carotene to be a good radical-trapping antioxidant under conditions of low partial pressure of oxygen, however. Now, in the lungs, we have high oxygen partial pressures expectedly. Okay? Now, in such environments, beta-carotene becomes an autocatalytic pro-oxidant rather than antioxidant, causing radical damage to lipids and proteins. Okay? Now, smoking, in turn, appears to increase the production of oxidized beta-carotene metabolites, thereby increasing the chances of developing lung cancer, and thereby tying up the results that we saw from the 2008 meta-analysis that was done. Now, similarly, with vitamins C and E, although a considerable body of epidemiological evidence suggests the protective effects of supplements, interventional trials have proved otherwise and have even found increase in mortality with certain types of cancers and cardiovascular diseases. Now, in the case of vitamin E, the relatively stable tocopheroxyl radical that is formed tend to persist long enough to penetrate deeper into lipoproteins and tissues. Now, this in effect causes further radical damage rather than interacting with a water-soluble antioxidant at that surface of the lipoproteins or membranes. Okay? Now, with vitamin C, on the other hand, although it can react with superoxide and hydroxyl radicals to yield monodihydroxyascorbate and hydrogen peroxide or water, it can paradoxically be a source as well of superoxide radicals by reaction with molecular oxygen or hydroxyl radicals in the presence of copper ions. Now, what is the take-home message from all of this? Unless there is a specific indication for using vitamin supplementation, it is best to avoid such supplementation given the current evidence. And after all, we get the right amount of vitamins A, C, and E we need from the diet alone. Okay? Now, furthermore, habits that promote inflammation and free radical formation, such as smoking, should naturally be avoided as much as possible. Now, this table here, which I got from the Harper's textbook, nicely summarizes the antioxidant and pro-oxidant roles of vitamin C. Now, I will end this video lecture on free radicals and oxygen toxicity here. You can now proceed to the next video lecture, which is on xenobiotics.